GMOs, genetically modified organisms. Misunderstood, proven safety and impact, critical tools for the future. Crop domestication and breeding has occurred for tens of thousands of years. Some examples are shown here of different products that came from wild relatives. On the left are brassicas that produce cabbage, kale, cauliflower, broccoli, a number of other types of things that came from wild mustard plants. Many of the vegetables and fruits that we enjoy came from wild relatives that had very small and a limited number of fruits, and now have much better tasting and larger and fruits that are free of compounds and non-nutritional compounds. A number of the grain crops that we have were domesticated from wild ancestors and selected for a high level of productivity within our systems. And things like the banana below came from wild relatives that were very seedy and hard to eat, and now we have the banana that we eat today that's very tasty and doesn't have those seeds. Animals were also domesticated from wild relatives. There's some examples here of the chicken, both in terms of the high level of egg production relative to wild relatives and in terms of the meat production. Those advances have both been important in feeding people, but also have resulted in a very high degree of feed efficiency and therefore reduce the environmental input related to food production. If we look at a timeline of genetic modifications, as early as 30,000 years ago, people began to domesticate wolves into companion animals. Somewhere around 7,000 to 10,000 years ago, some of the first crops were domesticated as civilizations began to develop. And it was in the mid-1970s that people learned how to clone DNA and produce modified organisms from that. Some of the first work was by Boyer and Cohen in 1973. And you can see a timeline of events that occurred, both scientific timeline as well as things like the Supreme Court allowing patenting of genetically modified organisms. Some of the first organisms to come out, the first crops to come out, included the flavor saver tomatoes, pesticide producing crops like corn and cotton that had Bt, herbicide resistant crops in 1996. And then there's been a number of potential products that have been developed, such as golden rice, that never reached the market. So a little bit on terminology. The term GMO, or genetically modified organism, first was introduced by the New York Times, so it's not a scientific term, but came out in the popular press to broadly describe organisms, whether crops or animals, that had some sort of genetic modification, usually through biotechnology. As I described earlier, genetic modifications and genetic selection has occurred for a very long time, but GMO more specifically relates to forms of crop genetic engineering, or what's often called crop biotechnology. There's a range of modifications. I won't talk much about the details of those, but some of the categories are transgenic, where the piece of DNA that's introduced into an organism has come from a different species. Cisgenic, where you're taking a piece of DNA from one plant and putting it into another plant. So maybe you take a disease resistance gene from an apple and put it into another apple variety that's susceptible to that disease. And then more recently, there's been a lot of discussion of gene editing. In addition to these types of modifications, there's other types of new technologies. And so one term that's being used more recently to encompass a broad range of biotech-related technologies in breeding is new breeding technologies. There's been lots of controversy on GMOs, often non-scientific, often related to people's opinions on intellectual property, on large companies, and various other sorts of things. And this is one example of a project that is very misleading, in my opinion, where things are labeled as non-GMO verified. And on their website for this non-GMO project, they list these different risk levels that sound very scary. So you have high risk, low risk, non-risk, monitor risk. And the risk is not related to the product itself or the danger to humans or animals or anything like that, but just the probability that you might have a genetically modified product derived from that. And it's extended so far to labeling things such as salt, which could never be genetically modified, as non-GMO, confusing the consumer in terms of the validity of this technology, and has a lot of people concerned. So I have a little clip from Jimmy Kimmel where he interviewed a number of people about GMOs, and I think it's just a little bit of a funny demonstration of the amount of confusion out there where people believe this technology is bad for one reason or another, but many people really don't understand the technology.
So, as I usually do when people take a stance on a complicated issue, I wondered how many people who are against GMOs really know what they are. So we sent a crew to one of our local farmers markets to ask people why they avoid GMOs, and more specifically, what the letters GMO stand for. Enjoy.
Do you try to avoid GMOs in your diet?
I do. Yeah.
Tell me why.
There's just a vibration with GMOs. For me personally, just something that I don't particularly want to put into my body.
What does GMO stand for?
Genetically modified. Genetically modified.
The O?
The
O.
I don't know.
Bishop Don Magic Wand, what is a GMO?
Well, I'm not really sure. You have to make me familiar with that. What is a GMO?
Do you try to avoid GMOs in your diet?
Yeah, absolutely.
Why is that?
Just the effects, I guess, on myself.
What does GMO stand for?
Man, putting me on the under the grill. Let's see. I don't even remember.
Do you try to avoid GMOs in your diet?
Absolutely,
Why?
because they're not good for you.
What is a GMO?
It's genetically I don't know, what is it?
If you are eating whole foods, you want to eat what you're eating. You know what I mean? You want to eat what you see, and you're avoiding processed food. But when the whole food is somehow contaminated, that's kind of making it a moot point.
What does GMO stand for?
Genetically manufactured. Oh.
What is a GMO?
General modified ingredient, right?
What is a GMO?
It's something modified.
Do you try to avoid GMOs?
Sometimes. Not a whole lot, but, I try.
What is a GMO?
I don't know. I swear. I was just going along with it. I don't know what GMO is.
What is a GMO?
I don't know. I know it's like some corn bad stuff, right? I know it's bad, but to be completely honest with you, I have no idea.
What is a GMO?
I don't know, so I really don't care. It doesn't affect me. I'm not sick. I'm fine. So.
What is a GMO?
It's a genetically modified organism.
And what's GMO spelled backwards?
OMG. Nice.
One of the first genetically engineered crops that saw extensive use was genetically engineered papaya with resistance to Papaya Ring Spot Virus. This was a devastating virus in Hawaii that basically wiped out the papaya industry, and this particular genetically modified crop had resistance due to the transgene that was in it, and was able to save that industry. So much of the papaya that is eaten these days in Hawaii is of this genetically engineered type. More recently, a genetically modified chestnut has been developed. This chestnut has resistance to chestnut blight. This is a disease that wiped out much of the chestnuts throughout the United States and is now under a comment period in terms of its potential to be released into the wild as a new chestnut variety and help hopefully to restore some of the chestnuts that were lost relative to the number that were here many years ago. Some of the other biotech crops that have seen widespread use throughout a number of the countries in the world had insect or herbicide resistance. So shown here are some examples of BT insect resistant cotton and BT insect resistant maize. There's also genetically modified soybeans and maize and cotton that are resistant to herbicides such as Roundup herbicide. There's a paper, I give you the reference here, Brooks and Barfoot, that describes some of the impacts of these genetically modified crops in the period from 1996 to 2016. These crops, the insect-resistant crops, have reduced pesticide spraying by 171.4 million kilograms due to their resistance to some major insect pests. With the BT corn, for instance, there's been a very large reduction in the European corn borer, and this has also allowed better production of organic corn because these insects just aren't as present on the landscape as they were before the BT corn. Some of the herbicide-resistant crops, such as the Roundup Ready crops, supported changes in tillage practices, resulting in reduced field use and resulting in facilitating tillage practices such as no-till and reduced-till. And the estimate in this paper is this reduction in greenhouse gas emissions due to these herbicide-resistant crops in 2016 alone was equivalent to removing 16.7 million cars from the road. So there are some very important benefits of the biotech crops in addition to some of the benefits to the farmers. As I said, there's crops that have been developed that have not been able to be marketed due to intellectual property and regulatory reasons. One of these is golden rice. And this golden rice was developed because it had a higher amount of beta-carotene in the grain. And this beta-carotene has the potential to help people who have diets that are low in this particular vitamin, resulting in blindness and sometimes death, especially in children. So the lack of access to technology, in this case golden rice is an example, but other technologies, does have a negative impact on people throughout the world in terms of food availability, the cost of food, and the nutritive ability of those foods. And so decisions not to allow GMO products can have negative impacts. A recent technology that's been discussed quite a bit is gene editing. CRISPR is one of the words that's used to describe gene editing. There's various approaches to it, but this CRISPR-Cas9 system is one of the most widely used and one that's seen very rapid growth recently. This is just a little bit of slide. There's different ways that plants can be modified. Mutation breeding is something that's been done since the 1940s when there was research on mutations due to chemicals and radiation, for example. In this case, you get many small random mutations throughout the genome. In some cases, some of those mutations can be useful. With genetic engineering, DNA is inserted into the plant genome. So now looking at the left of this slide, that DNA occurs randomly in random places throughout the genome. So each time you put that DNA in, it goes into a different place in the genome. But at least in this case, you know that there is a small piece of DNA that's put in with a very specific function, and that function can be characterized. CRISPR is something that is an advance over both of those technologies in that you can make modifications in the genome in a very precise location. In some cases, changing in a targeted way as small as one base pair or 1/billionth of a plant genome, for instance. So it is an advance in terms of the precision and the types of modifications that can be made. In some cases, it can be used to introduce DNA, but in a number of cases, it's used to just modify the sequence of existing genes in the genome. Some examples of the types of products that have come out of this is the CRISPR non-browning mushroom. It was one of the first demonstrations of this particular technology. Waxy corn is a type of corn that's been useful for animal feed. It's a little more digestible by animals. The types of waxy corn that had been used before were due to naturally occurring mutations. Using CRISPR and gene editing, in this case the company Corteva was able to reproduce that waxy mutation in a very targeted way in new lines of corn. So it's a way to very precisely introduce a specific mutation in lines of corn that didn't previously have it. Through breeding, you could introduce the mutation, but then you'd also have hundreds of genes that are nearby that mutation that have other effects on the genome that are in. Here's an example of gene editing in wheat. Wheat is a polyploid species, so it has 3 copies of each genome. In this case, a gene had to be knocked out or disabled in all 3 copies of the genome, something that would be very difficult to do with mutation breeding or regular breeding. And when all 3 copies of that gene was knocked out, it gave resistance to this fungus powdery mildew. So it's an example of the precision and power of gene editing in a complex species like wheat. One of the more recent products that's come out is high oleic soybean. This is another example of a product that there was a similar type of product that had been developed using mutation breeding, again with other mutations in the genome, and by gene editing, Calyxt was now able to develop a edited soybean that had the same high oleic type of oil content as the previous soy, and so they reproduce that mutation in a very precise way using gene editing. A lot of focus these days is on producing healthier and more nutritious foods. One of the complaints about the use of biotechnology in the past is that the products had benefited the farmer and had little benefit to the consumer, separate from lower price of food and high quality food, but the consumers didn't see necessarily a direct effect. And so there's lots of examples now where people are trying to use gene editing to eliminate the seeds from fruits or to increase the nutritional concentration of fruits or vegetables and other products like that. And those are expected to be in the market in the next few years. I described to you the domestication process and the development of new crops from wild relatives. This is something that in the past occurred over thousands of years, sometimes 10,000 years to get to the types of crops we have today. But there's a big interest in having a broader diversity of crops to be used in our cropping systems, and there's potential that with our increased knowledge of the genome and gene function that we might be able to use gene editing to more rapidly develop new crops that can enter into the cropping system. Another example of using, in this case, a cisgenic technology where a potato gene is reintroduced into potato is this innate variety by Simplot that has reduced browning relative to other potatoes, and transgenics have also been used to introduce disease resistance into potatoes, reducing the amount of spray needed. Gene editing is also used in animals, so one of the examples is gene editing in pigs that made them resistant to a specific disease that was quite a problem in pigs, and so this is one example of a benefit that should also have a substantial reduction in the cost of pork production and certainly improve the lives of the pigs that are produced for meat. Another example of gene editing in pigs, which I thought was quite interesting, was this switching off of pig viruses that are in the genome, and that allowed the pigs to serve as a source for human organ transplants. So pigs and humans are relatively similar in their physiology. There's been a strong desire to be able to find more organ transplant donors, and so this is a gene editing project that led to the potential to more easily use pig organs in humans. And one of the reasons that this was interesting is that there's a discussion that with self-driving cars there will be a substantial increase in safety, and that will lead to organ shortages. And so it's quite interesting to me when you look at the whole system of things. You have one technology that might increase the safety of that particular technology, but have another substantial impact that was maybe unexpected as that technology was developed. So there's lots of things when you look across entire systems in terms of the impacts of one technology on other outcomes. So just in summary, This is a little bit of a summary of how things have gone. If you like the software designation of various versions, starting as much as 10,000 years ago, much of the selection was due to unconscious selection. People just saved seed from the best plants and carried it over from year to year, or saved those plants in the environment if they were perennials. And then over time, people began to be more systematic, even before the principles of genetics were understood, and so on. People were doing conscious selection. And then it was in the early 1900s, now a little more than 100 years ago, where formalized plant breeding, plant and animal breeding, really was occurring at a much higher frequency. In the early 80s, we started to use genetic markers to improve the process. In the early 1990s, genetic transformation, so I showed you some examples of that. And then we moved to genomic selection, and more recently now moving to the widespread use of genome editing, hopefully to develop new products. And so these technologies are part of an ongoing continuum that should be very helpful in being able to advance both annual and perennial crops and new types of production systems. And I think they're going to be very important technologies for the future.